The disease I'll be talking about today is called the von Hippel-Lindau disease, also known as the VHL disease. This disease is an inherited disorder discovered by the German ophthalmologist Eugen von Hippel. The most notable characteristics of the VHL disease are the formation of tumors, predominantly hemangioblastomas, which are tumors in the central nervous system, and fluid-filled sacs in various parts of the body, such as the retina, which leads to blindness. What causes the VHL disease is the mutation of the von Hippel-Lindau tumor suppressor gene on the short arm of the chromosome 3. The gene encodes for the VHL protein, which is involved in regulating the degradation of another protein called hypoxia-induced factor 1 alpha, HIF-1 alpha. It does this by adding ubiquitin groups to the HIF-1 alpha, which is then degraded by proteasomes. Mutations lead to the production of faulty VHL proteins, and these faulty proteins are unable to effectively regulate the cell cycle, resulting in uncontrollable cell growth and division that leads to formation of tumors and cysts. VHL disease is inherited in a dominant pattern, which means that one copy of a mutated VHL gene is enough to cause the disease.
One experiment we could do to examine the function of the VHL protein is to create a homozygous knockout of the VHL gene and see what happens to the HIF-1 alpha activities. I would use the zebrafish as a model organism because the HIF pathway is highly conserved, and unlike zebrafish, the VHL knockout mice obtained by homologous recombination die in utero, while zebrafish mutants can survive until larvae stage. I would use reverse genetics to obtain homozygous knockout mutants. Taking E and U mutagenized adult males, I cross them with wild-type females and then prepare a DNA library of tail fin clips from F1 males. Use PCR to amplify exons of our gene of interest, and then sequencing is performed on the genetic library. Once the mutation is identified, the fish carrying the mutation will be crossed with a wild-type fish. The resulting progeny, F2, are then intercrossed to generate F3, which will have homozygous mutants for the VHL gene. Then we can determine whether the HIF-1 alpha target genes are upregulated in VHL mutants by performing a microarray using mRNA from the mutants, and gene expression can be verified using whole mount in situ hybridization.